I'm a mother of 11 in total. I lost my husband 6 years ago to cancer, and I lost my parents 13 years ago to cancer. 2 years ago I was diagnosed with my first mammogram, and I went through all the treatment, all the chemo, all the radiotherapy, and I've come out the other side, which is why I'm throwing myself into raising money for cancer research. It's fantastic to hear that Professor Hannon is willing to do so much research into this. Obviously it's going to be a lot of money, but somebody is willing to give up so much to actually go and research so much into this horrible disease.
Cancer is one of the biggest challenges facing our ageing population, and we're here at Cancer Research UK's Cambridge Institute to meet with Professor Hannon and his research team to find out what they're doing to beat cancer quicker. So hello, Professor Hannon, thank you for joining us. I was wondering, could you tell us a bit about yourself?
Well, I'm an American. I spent 23 years working at a place called Cold Spring Harbor Lab in New York, and then decided to move to the UK 2 years ago.
So why did you come to the UK?
Well, I loved Cold Spring Harbor, spent 23 very happy and productive years there, but it was time for a change. It was time for new colleagues, new challenges, new adventures. And so part of the move was creating a smaller, much more focused research group, and one where I kind of had the fun of science again, the fun of interacting with the students and postdocs in the lab, planning projects and this sort of thing. Also the CRUK Cambridge Institute has phenomenal colleagues, great resources. CRUK was really terrific, extremely supportive during the move, and so that sort of, there was a whole set of draws to CRUK, and a sense that it was time to do something different, to move on from Cold Spring Harbor.
And so what is your research here?
We have about 4 different research areas. We're a little bit broader than many labs. So obviously we work on cancer, being here at the CRUK Institute, mainly on breast cancer, though we have some small side projects, usually collaborative, in other tumor types. We have a strong interest in epigenetics, and that kind of bridges also to interest in non-coding RNAs. We have a strong, we've always had a strong focus on technology development. And then we work on sort of systems which which protect germ cells and protect germ cell genomes, protect the fidelity of transmission of genetic information from one generation to another.
Hello, so my name is Bruno. I'm a research associate here at Greg's lab, and I'm trying to make the biological models to which we're going to use with this big 3D tumor project.
So we've just been talking to Greg Hannon about his research.
Okay.
So how did you end up here in this lab?
Well, I finished my PhD about a year ago, and I wanted to pursue a postdoc, something a lot more translational, and I knew that Greg was doing something really cool about the cancer and developing new techniques. So I contacted him, and we got along well, so here I am.
So what is the research that you do here?
So I'm part of this big project, the 3D tumor that we're working on, and I'm trying to make mouse models to be able to observe what kind of cells are going into these tumors. So parts, besides what Greg probably already told you about the pathology and the way you can actually make virtual pathology, I'm also interested in part of the research, and I want to know what kind of cells from the immune system get infiltrated into the tumors. So part of the project will be taking these biopsies from patients, put them in a mouse model that we call the patient-derived xenografts, and then I need to observe, or I need to have a tool to be able to observe what kind of cells from the little mouse are going to inside the tumor that sometimes either help the tumor to spread or actually help fighting the tumor. And for that, we need to label with some kind of colors and fluorescent proteins these kind of cells.
So you end up with a model of a tumor, but it's in a mouse, and you can actually watch how the body fights that tumor.
Yes, pretty much.
So what are the biggest challenges you've had in trying to do that?
So far now it's just cloning, and so basically we are trying to make gluing all of these different genes that I need to be expressed on this mouse. And that's by itself is a challenge, because you want to each of these mini genes to act independently and not have the influences from another ones. And for that, we use this kind of specific linkers in between them, which sometimes complicates what might be theoretically easy. It's not that nice. So these last 4 months, I've been trying to just to make this big construct, so gluing all of these genes together.
And what is going to be the impact of this work?
Now we can actually have a real-time observation of what's going on. And if you're talking about the impact of grand scale of all of this project, you can actually, now you're able to merge different kind of data that you usually acquire independently. You can actually merge in the whole module. And then you have the ability to see it as a single object. And that will allow you to search for patterns that were not easily pinpointed if you just look at this data all by itself.
So what is the greatest challenge you've faced so far?
I mean, that's a hard one. I think, all of us are faced with a series of challenges which generally are technical in nature. We know the questions we want to ask. The mechanism to get to the answers is often encounters a technological barrier. And I think one of the reasons why the lab has such a strong focus in technology development, and always has, is trying to get to those endpoints. We would hit something where there wasn't a way to do what we wanted to do. And so then we would end up developing something like XM capture, which we did a number of years ago, or ways to make really complex oligonucleotide libraries for cheap, basically. That was a challenge, taking something that would have cost $1 million and making it cost $5,000, right? So those are, I think that's a generality in research.
So in relation to the Grand Challenge and your work for that, we've heard that you are in the final stage of being shortlisted. So where has the challenges come for your Grand Challenge proposal?
We were shortlisted for the Grand Challenge. We're one of 9 teams. It's unclear whether they'll give 1 or 2 awards. We've submitted our final application, this incredible tome that we had to put together, and we go for a kind of a pitch and in-person interview in mid-September. So there were a number of different Grand Challenge areas. Ours was in it was called tumors in 3D. So we'd been thinking for a number of years about the fact that, I don't think anybody really knows what a tumor looks like, right? So we see slices of these things in pathological specimens, all the time. Sometimes they get molecularly annotated to a small degree, individual protein expression, something like this, right? So a number of years ago, I thought, well, wouldn't it be nice if we knew what a tumor looked like? And then as we started discussing the Grand Challenge project, we sort of expanded that idea and said, okay, we would like to know what a tumor looks like, what all the cells in the sample are, what are their familial relationships to each other, what are their gene expression states, and how are they affecting each other. So if you think about it, it's sort of like a Google Maps for tumors, but one where you could click on a house, who's home, who's on their cell phone, and then what they're saying to the neighbor. And so the idea is to get this very, very detailed idea, snapshot of what's happening in a whole tumor at once.
Hi Dario.
Hello.
So could you tell us a little bit about yourself?
Sure. So I've been in the laboratory of Greg for quite a long time. This is my 7th year, I think. So before we were in the US in Cold Spring Harbor, which is where I did my PhD after moving from Italy. And then we were running out of a project that I was doing, and this was sort of just starting. We had some idea about measuring gene expression directly on tissues. It wasn't really formed to the point it is now. So that is essentially not just my research, that's kind of one of the big points of this entire project. The fact that you want to know what the tumor is doing. And this is mostly done by knowing which genes the tumor is switching on and off, because from there you can derive which pathways are active, how it behaves, which drugs it may become resistant to, and so on. But the problem is that so far this has been mostly done at the level of either groups of cells or biopsies or even the entire disease. What we think is needed is to do it at the level of every single cell of the tumor, and not just in isolation, but looking at them where they live. We're going to find them in their home. Just going to go there, look at which genes are on or off. So what I'm doing specifically here is a couple of things. First, I'm trying to optimize and improve on a beautiful system that's been developed by one of our collaborators, Xiaowei Zhang in Boston. And this essentially allows you to assign a zip code to each gene in the cell, so that rather than having to image them one by one on a microscope, you can just, it's the same principle essentially in how a 6-digit postcode can tell you about the place where each house in UK is. This way we can know where each gene is and image it just by doing 6 rounds of imaging rather than 3,000, for instance.
So what are the challenges you face trying to do this?
The biggest challenge is that we need to image across a lot of different scales. You imagine a tumor is a centimeter in size. A cell is 10,000th of a millimeter in size. We need to move from different type of microscopes continuously in order to image across these different scales. How do you know that you're looking at the same cell? How do you move from a super magnifying lens to a wide angle and know you're looking at the same thing? So what we are trying to develop, what I spent quite a bit of time now, is try to develop a series of fiducial marks, something that you can image across all the instruments of your technique, all the instruments used in the project, and you can cross-reference them across the data in order to be able to keep this registration across scales. That's one, and the other is just purely coordination, because we're talking about 16 groups, sending samples from one group to the other, trying to make sure that the tweaks that you do to your technique don't necessarily impact what's happening to the other techniques. And that's why we are not doing this as a consortium of groups that somehow talk to each other. We are literally mailing to each other every day. It's been accelerating.
So communication is a massive, massive part of your work.
Which is why I spend now probably 40% of my time in front of a computer and the other 60 is in front of the bench. But that's needed, just making sure that the analysis is online.
So what is your hope that this work will do for our understanding of cancer?
I think this could be potentially quite massive, because essentially if this works, if the gene expression part works, in one experiment we are gonna sequence, we're gonna get more gene expression information that the collective humanity has done so far, pretty much. We are drafting in computer scientists and even game developers from virtual reality, so just making sure that they can exactly interpret your data the way it is, so that when we give the data set to the doctors, to the people that actually have to take decisions that involve people's life, they don't have to bother about is this gene in this cell or this other cell. We can sort of give them something where they don't even notice how much work was done before. If they don't notice that, we win. They can just use it to take decisions. So the challenge then is going to be interpreting this data, but potentially you are moving from a scenario in which you have like 40 people in a room and you know that 5 of them are going to die from the disease, but you don't know which one they are and you don't know how. From you won't tell people you've got breast cancer of this subtype. You're going to tell people your specific one has this feature and we've got these 10 cells in this area of the tumor that are showing this resistance. This is exactly how we are going to go and hit them.
So a huge amount of information.
Yes. Trying to go big data, but not just a list of data that is tied to a map, which tells us exactly where it's happening, where things are happening, so that we know exactly where to go to try to influence them.
So what's your hope that this technique will help, and how would you think it will help us in fighting cancer?
Well, it will give you another way of doing diagnostics, for example. You as a pathologist, you'll no longer need to be on the lab doing all of this wet lab work to see all of your tests. You just have this model already on the computer and you can quicker and easily do it. And besides that, you also can, since it's a 3D, you can actually see it on from different angles and actually have much more control on what you're doing. At least, and as Greg says, we are kind of transform pathology in the way pathology is made.
And what would be the impact of us actually understanding that level of detail and being able to see that level of detail in a tumour?
I mean, the truth is we don't know. We're walking into this, that level of information has never been available. But what we do know is we can apply the most sophisticated genomics to patients. And we can, to some degree, predict the course of their disease and what treatments they'll respond to. But those predictions are very imperfect. Better for some tumor types than others, but by and large very imperfect. And so for patients that present with, essentially indistinguishable disease, we think that encoded within, this higher dimensional information, something that takes into account not just its tumor type X, but here's what all the cells in the tumor are like. Here's how it's interacting with its host. We think it's somewhere within that information will be what's needed to better predict course of disease, better focus treatments.
So the actual environment and the shape and all those things about the tumour we normally ignore could be very important, but we need to look at it to find out.
Right. And I mean, it could be, it could be transformational even far beyond that. That's sort of a proximate goal. Taking this information, correlating it with clinical data, and seeing, what features in a patient's tumor correlate with how that patient performs. But just even from the basic biology standpoint, how tumors evolve, I think that we could learn a tremendous amount. But nobody's ever had this kind of information, so until we do it, we don't know.
So it has fantastic potential, but how does the Grand Challenge help you do that?
Well, the truth is this is a very high-risk, very innovative project that requires a lot of money. And so, the thing about the Grand Challenge is, they're looking for innovation, they're looking for very high impact, they're looking for people to go take risks. And so, for us, to conceptualize a project on this scale requires a very big financial investment, because we can't do this just as our own lab. We need people who have, expertise. We have collaborators in chemistry, we have collaborators in astronomy, we have computational collaborators, we have people who are experts on the clinical disease, we have people who are experts on tumor-host interface. We have to bring all of these guys together. In fact, we even have a virtual reality game designer as part of the team, because we have to present the information somehow. And, sort of bringing the best people, the people who've invented the measurement technologies that we're going to use, getting them all onto one team needs a scope of funding that is like the scope of the Grand Challenge. And in fact, one of the sort of driving forces, as I understand it from the CRUK, behind designing this funding mechanism was to give people the resources to bring together these very high quality international teams.
So if there is this situation where you need quite large sums of money and they're very competitive projects, what does that competition feel like to you? Are these other people's projects phenomenal from your perspective, or do you not even get to see them?
We get to see a title in an abstract, so we don't know much more beyond that. And the truth is they're spread across a number of different categories. There are 3 teams competing in our category. But the truth is I haven't paid that much attention to it, because the timelines here have been very short. So, we got the, here's what the topics are. We put in the preliminary application, and then it's a very, very quick run-up to the final application, which was extensive than the interview. So we haven't really had time to do anything but focus on, making our project the best that it could be.
So what do you think the chances are for anyone at this stage?
Mathematically, 1 in 9.
1 in 9.
All I can say is that we put a lot of effort into this, and for us, our excitement in the project has built over time. As we brought our collaborators together, we had a meeting at Trinity College in May. Interactions amongst the team have been incredibly energizing. So I think that's a good sign.
So even the process itself of getting to the grant has taught you a huge amount.
Enormous amount, and bringing, there's one thing for me to have an idea. I think I had, after climbing 2,000 feet on the bike and was incredibly hypoxic, and then it sounded good. But it has continued to sound good, and the more people that we bring in, the more it not only sounds like a great idea, but also sounds possible. And so, of course, in science, you're not aware of everything that goes on, but then I started looking at these in situ measurement technologies, and brought in collaborators that could do things that I didn't even know were possible when I was kind of envisioning this thing as a whole.
So if unfortunately you didn't get the funding,
Yeah.
do you still think there's somewhere for you to go with all this work?
No, I think everybody who is involved is very committed to trying to do this. And so we've run for 6 months, kind of with each person donating bits and pieces from some discretionary resources. But we've really come actually quite a long way to proving to ourselves that this is feasible. And we actually was up in Dublin last week. We have a VR viewer built where you can, something we call Superman mode, literally fly through, in this case, a tumor model, and see the different cell types. And you can point to a cell, and the simulation will tell you what the cell is, and it's really fantastic.
movies.
Yeah, and in fact, that happened with 3 people part-time for just a few months. So I can't imagine if we have them full-time for 5 or 6 years, what would be possible.
So if you don't get the funding, this is going to happen, but the money will
It'll
make a
be
huge
it'll be
difference.
drip-fed,
Be drip.
and we'll have to chip away at individual aspects of this one at a time. We may not benefit from this sort of synergy and critical mass. Now, our vision of this is like kind of a Manhattan Project, which is, we don't put money into a whole bunch of individual labs, but the group gets together, hires a set of people who work in one place. We put them in a room, we bolt the door and say, come out when you're done. But I think that this works best if we have the astronomers sitting next to the computational biologists, sitting next to the people who are dealing with the clinical samples, who are dealing with the mouse models, so that they all talk to each other.
And that's what
That's the goal. That's what a grand challenge would do for us. That would be completely impossible to do in another way with another funding mechanism.
My role is just a fundraiser. I like to raise money for them as much as I can. I'm involved with the Time Bank, which raise money for cancer research every year. We do a big fundraiser. We raise £1,100 each year. I do the Race for Life. I've done that twice now. And any other thing that cancer research want me to front, then I will do that as well. We did a big campaign for saying thank you to cancer research in their shop. And myself and my children went and signed a big card and had a photo shoot with them, just to raise awareness of people saying thank you. I love being a volunteer. It's brilliant. It's so rewarding. Anything else, you just get paid for it and it's just your job. Whereas volunteering is just so much more rewarding. I volunteer, as I say, with cancer research, with the Time Bank, with the Food Bank. There's just so much more to be had from that. So I was invited to go on stage at the Race for Life this year, and I had to stand up and tell my story in front of 5,000 people. Never ever done anything like that before, but it was lovely. It was just such a fantastic response from the crowd, because they were doing exactly the same thing. Everybody somewhere has been touched by cancer, and to know that we are raising so much money to cure it, it's amazing. And I think the only challenge we had was the fact that the fundraiser we did last year, we found out that actually the amount of people came, the room wasn't big enough. So apart from that, no, there's been no challenges at all. As far as I'm aware, most of the money goes on researching for the disease, and we need to get this disease stamped out as soon as possible. But obviously some of the money, I presume, must go towards the advertising for fundraisers, because obviously the staff are going to need paying and everything, so there must be a certain proportion of that goes there. But I'm hoping that most of the money does go towards beating this disease. I'm of the opinion that the government should put more money into fundraising for cancer research. I don't think they should have to do it all by themselves, but it's, as I say, it's something that touches everybody, and we need to get as much research done as possible and as soon as possible to save more lives. Anyway at all. There's cancer research shops, you can go online, you can do the Race for Life, there's Tough Mudder, there's just so many ways you can raise money, and you can hold your own fundraisers and donate it yourself. Everybody should get involved. It's so rewarding. We need to beat this. We need to beat cancer.